Hello, students. We're going to continue on and look at the three-dimensional structure of proteins. So there are four kinds of protein structures: primary, secondary, tertiary, and then some proteins have what is called quaternary. We're going to go through each one of them. The first is primary structure. It refers to the amino acid sequence. So if you see here, there's the letter designations for the amino acids, and it's the order of the amino acids. Normally it's written as from N terminus to C terminus. Secondary structure are regions within that show kind of a regular repeating structure. We're going to go into more detail on that. This one shown here is one of the most famous kind of secondary structure, which is alpha helix. Coming down here, once you get to tertiary structure, it's what I would call a well-defined folded structure, sort of the three-dimensional view of the folded protein. That can be usually spherical for things like enzymes, where it has an overall spherical shape. In proteins that are more structural, sometimes they can look more rope-like, but it's the three-dimensional structure after the amino acid chains have folded into their working structure. Some proteins are made up of multi-subunit complexes, and that would exhibit quaternary structure, where they have multiple polypeptide chains. And this is not all proteins, but the one that we're going to look at in detail next week is hemoglobin. Okay, so here's our primary structure. This is a amino acid chain. So we have kind of that repetitive sequence: N alpha carbon, C double bond O, N alpha carbon, C double bond O, N alpha carbon. So you can see all the alpha carbons there. This shows them in a more tetrahedral shape around the alpha carbon. It also shows the different R groups. So here's R group 1, R group 2, R group 3, R group 4, R group 5. So we're starting from the N terminus to the C terminus. Okay. Secondary structure is that amino acid sequence can sometimes form structures that are recognizable and similar to each other. The two most famous are alpha helix and beta pleated sheets. And then there's a third, which are like turns and loops. Now, this is a great time to tell you that this particular picture is showing alpha helix and beta sheets. The interesting point to be made here is that they look like a ribbon and arrows, but anytime you see these structures, I want you to think about all the amino acids that we've been talking about and their properties. Okay, so they're not just these blob structures. So alpha helix, probably one of the most famous kind, is a coiled structure, and it's stabilized by hydrogen bonding. The main chain, C double bond O, will hydrogen bond with NH groups within the chain. And we can kind of see here N alpha carbon, C double bond O. So anything blue here are nitrogen. So N is blue. The alpha carbon is black. The kind of red pink is the oxygen attached. Should I say pink or red? Okay, it's just to get you to kind of look at the structure. It forms a coiled structure. Every 3.6 residues, and the word residue is going to be a new word for us to get used to. That will be the amino acids. Every 3.6 residues, there's kind of a bend. And that's why they use the ribbon, because the ribbon's a good way of thinking about it bending. Okay, so if you look here, I'll try to kind of pick one in the middle of the structure. So here's N, C, C double bond O, N, C, C double bond O, N, C, C double bond O. And then what you'll also notice, if you look carefully at the structure then, every 3.6 R groups, it has that curve to it. The curve is said to be right-handed. So if you hold out your right hand and thumb and you point it, you'll see that it points this direction. So it curves like that. Okay. All right. So let's take a look at some other pictures. In this case, this is trying to get you to think about how it's curving. So here we have N, C, C double bond O, N, C, C double bond O. So here's our R group 1, R group 2, R group 3, R group 4, and then it curves now. And so there's hydrogen bonding between this C double bond O and this NH. And that's shown here by these dashed lines. That's the hydrogen bonding. So hydrogen bonding is going to be one of the most important ways that biochemical structures are stabilized. The other thing too is if you picture that ribbon structure, if you picture that you're looking down, so you're going to look down, it's going to actually have kind of this hole here. And so this structure is this one, but you looking at it, looking down on it like a cylinder. Okay, once again, take another look of the curvature every 3.6 residues, and residues are these R group amino acids, 1, 2, 3, 4, but it's more like 3.6. The other thing that happens that's really important is, let's take a look. These are the alpha carbons that I'll highlight. Do you notice the green? The green, this is the R groups. These are the R. So do you notice in this model here with the ribbon, do you see the R groups? They point out from the backbone. So the ribbon is the backbone, and the backbone is NC, C double bond O, NC, C double bond O, and sticking out from that backbone are the R groups. Okay? So alpha helices are a very important secondary structure in biochemistry. And anytime we see like the ribbon structures in a protein, you're going to think of this structure. Okay, so here's another view of that. Okay, and then again, here's that ribbon. One of my big things, though, is to not see things as just blobs and ribbons, that this particular structure is just there for simplicity, but it's actually all the amino acid residues that we've been talking about and the polypeptide chain. Second important secondary structure, so we're still on secondary structure, are beta pleated sheets. They are 2 or more beta strands hydrogen bonded to each other through the backbone, C double bond O and NH groups. So there's 2 types. There's what's called anti-parallel beta sheets, and then there are parallel. So in this case, let's take a look. So remember the blue here, N, C, C double bond O, N, C, C double bond O. Okay. And then do you see how on this side it's the other way? N, C, C double bond O, N, C, C double bond O. And those are strands that are running in opposite directions. And so that is said to be anti-parallel because this one's running N to C and this one's running N to C. And what's holding them together are hydrogen bonding, right? Here you have the C double bond O, hydrogen bonding to the NH group here. And on this one, it'd be the NH hydrogen bonding to the C double bond O. And this is going to keep the two strands in a secondary structure. It also creates essentially what's called more like a sheet, right? And you'll see that in the next pictures coming up. These are parallel. These are adjacent beta strands that run in the same direction. So N, C, C double bond O. So this is N to C. And in this case, N to C as well. And they're stabilized again by hydrogen bonding. Okay. Many beta strands, usually 4 to 10, can come together to form large beta pleated sheets. And beta sheets can be purely parallel, purely anti-parallel, or a mixed composition. And there's going to be different representations. They can be flat or they can be twisted. And these big giant arrows are going to be the simple way of writing beta plated sheets. So for example, the direction of the arrow is indicated, the N to C polarity of the beta strand. And so if you notice here, these arrows are in opposite directions. So those would be anti-parallel, and these are in the same direction. So those would be parallel. Okay. Some proteins that we'll see will favor alpha helices and some will favor beta pleated sheets. It really depends. And we're going to see more of this as we keep moving forward. So this gives you an example of a fatty acid binding protein that is rich in beta pleated sheets, but we will come back to this, and it's something that as we look at more proteins along the way, it's just being able to interpret what we're looking at. One last thing is that these kinds of secondary structures are linked together by what are called loops. And so you can see these loops here. There's all sorts of different kinds of loops. I don't really ask questions about this, so I guess you can ignore this slide. But I just want you to be aware that if you notice here, you have these beta pleated sheets, the arrows, these are anti-parallel, but they're connected through these loop structures, okay? Okay. One thing you should note is there is a huge relationship between the structure of a protein and its function. So there are what are called fibrous proteins. These are examples would be like keratin, which are hair, fingernails, collagen, which is abundant in connective tissue. And so the role of let's say collagen as a connective tissue is for strength component. And so the type of protein structures that they have tend to be fibrous. They look like rope structures. So going forward, this particular structure here is worth noting, which is the structure of collagen. Collagen is what's called a triple helix. To distinguish it from the alpha helix, these are 3 separate polypeptide chains. They are highlighted over on the right side in 3 different colors. But again, what I'd like you to think when you see this big blob is that those are amino acid residues. Collagen has a very fascinating structure that dominates, and that is, let's take a look at the top there. Do you see something here? We have glycine, proline, methionine, glycine, proline, serine, glycine, proline, arginine. Do you see a trend? And the trend is that it tends to be glycine, proline, and another amino acid. If you look closely, here's that unusual ring structure for proline. And what's so interesting is that this has that intramolecular bond. And what will happen is that it tends to form sort of these crevices just by nature of the 3D that it forms. And those then provide the structure for it to interlock with other polypeptide chains to form the triple helix. So they can interlock with each other. So here's another collagen view. So it has the heavy dominance of glycine and proline, and then it forms this triple helix. And if you notice, then it forms a rope-like structure. And in fact, the electron microscope view of collagen is just that. And remember, collagen dominates in connective tissue, which kind of needs sort of that tensile strength, that strength to put to almost serve like a rope within our bodies. So this has definitely that look to it. This is going to be very different when compared to proteins that form spheres, okay, more spherical structures. Okay, tertiary structure defines the specific 3D shape of the protein. And a lot of them form this, which you look here is a 3-dimensional shape, is a what I would call a sphere. And what is so interesting and something we don't have time to go through in this course is that there's definitely a thermodynamics and energy discussion when proteins fold. There are scientists who study that process. Think about how amazing it is to go from an amino acid chain all the way, to a structure like this that has only one structure that's functional. So we're going all the way from an amino acid structure, sequence, I should say, primary, to secondary, to a tertiary, which is the 3D structure, which is now a functioning, fully functioning protein. It is so amazing that they have one 3D structure that will be functional for whatever the role of that protein is. So next week, we're going to talk more about myoglobin, which is shown here. Myoglobin is what is called an oxygen binding protein. And it's going to give us a good example of tertiary structure and function. And then we're also going to talk about hemoglobin, which shows quaternary structure. One of the first things I'd like you to think about, though, is when a protein folds, if the outside of the protein is going to be in contact with water, that the amino acid residues, okay, residues is the word we call an amino acid, R group, that residues that are on the outside of the protein would have to be hydrophilic. So ones like serine, right, that we would expect on this outside, something like serine, because it's in contact with water. Okay. Hydrophilic amino acids. On the interior, buried as it folds, on the interior would be nonpolar. Hydrophobic ones, like our alanine, for example, CH3, right? The nonpolar ones would be buried in the center of the protein. And this is something that I'm going to keep mentioning as we keep studying more and more proteins. Okay, myoglobin is a oxygen carrying protein, and it will have a very specialized group here shown in red, which is called a heme group. But for right now, what I'd like you to do is notice its 3D shape, and that what dominates in myoglobin are alpha helices. You see the ribbons? Okay, so what dominates in myoglobin are alpha helical structures. Okay, this is also showing you the distribution of hydrophobic and hydrophilic residues in myoglobin. And the hydrophobic ones will dominate within the interior of the molecule. The hydrophilic ones will dominate on the outside that are exposed to a polar solvent like water. And then shown here in these letter designations are the amino acid residues. So never forget, even when you're looking at the ribbon structures, that those are amino acids. And then definitely in myoglobin, if you notice, the secondary structure that dominates is alpha helices. Okay, in tertiary structures, there are a wide variety of structures that are stabilized by different types of interactions, some covalent and some just intermolecular. So in tertiary structure, hydrogen bonding will dominate between polar residues that allow for hydrogen bonding. You can have what are called salt bridges, which are electrostatic attractions. You can have hydrophobic interactions. And then you can have also covalent bonds, which are disulfide linkages. And then some proteins have actually metals in them. And so you can have metal ion coordination. Now, let me show you each of these in the following picture. This is one of my favorite slides. This is showing all the stabilizing structures, interactions that can stabilize 3D structures. So now your protein is folding, and it has to find a way to stabilize those amino acid interactions. This first one here, take a moment and take a look. This is these are 2 amino acids here, right? Amino acid serine, and it's hydrogen bonding to this other residue. Do you see how this has an OH group and this amino acid has the OH group? And so they can hydrogen bond. Okay, so over here, second structure, we have a positively charged amino acid and a negatively charged amino acid. This is what's called a salt bridge. This amino acid is lysine, and this amino acid is aspartate. Okay, so what is important there from our previous discussion is to understand that aspartic acid, that's its R group, It's going to be deprotonated based on its pKa, and so it has a negative charge. That's why we studied that in detail, is to understand that aspartic acid has lost its H, it's deprotonated, because this would be our discussion at pH 7.4. And so aspartic acid is really the ion aspartate, and that has a negative charge, and it can form essentially what is an ionic bond. Now, ionic bonds tend to be metal, non-metal, and there's no metals here. So in biochemistry, we tweak the name and call it a salt bridge. It's the interaction of a positively charged amino acid with a negatively charged one. Okay, let's keep going here. That now we have these two amino acids interacting. Can you determine what you think the amino acids would be as far as what properties they might have that might cause them to attract to one another? So if you said van der Waals or London dispersion, you're right. Those guys will attract via those forces, and they are both nonpolar. We would expect this to occur in what we would call the interior of a globular protein, right? Remember, those hydrophobic residues are buried in the interior. Okay, and then lastly, this would be an example of perhaps an enzyme. We'll see later, there's some enzymes that have metal ions, and that has a metal ion, and it's interacting with the carboxylate ion group, and they form an interaction between a positive ion on the magnesium and the negatively charged carboxylate group. Okay, that is glutamate there being shown. So that's our metal ion. But I think for right now, most important are these three stabilizing factors: hydrogen bonding, salt bridge, and van der Waals. These help stabilize the protein as it folds into its one three-dimensional shape that gives it its ability to function. Okay, this is just showing you that overall, when they fold, a lot of proteins have sort of that spherical shape I mentioned, but they're different, right? Like in this case, the first structure is myoglobin, heavy on alpha helices, secondary structure. The second protein there, that one has a lot of beta pleated sheets. And then the third one has a combination of both. This one's interesting. We'll come back to it. This is an example of an aquaporin, which is a protein that allows for the passage of water into the cell. And so this is a channel. So there are some proteins that serve as a channel to allow for the passage of something in and out of cells. We'll talk about this one more when we get to the discussion we'll have on cell membrane. Okay, quaternary is when you have different polypeptide chains, which are called subunits, and then they fit together to form a multi-subunit protein. And there's always a reason why it has a function. And our example that we're going to cover in more detail next week is hemoglobin. So hemoglobin has 4 subunits. Hemoglobin's function is to transport oxygen to our tissues. So we will come back to this as we get more in that depth, but quaternary structure, to know the definition, is when you have more than one protein subunit. More than one polypeptide chain subunits fitting together to form a multi-subunit protein.